Hello everyone and welcome to today's webcast, One Factor at a Time, supporting ADC process characterization and product specification. I'm Tim Wright, editor of Contract Pharma, and our webcast today is being sponsored by Piramal Pharma Solutions, a CDMO offering end-to-end development and manufacturing solutions across the drug life cycle. Pyramil serves clients through a globally integrated network of facilities in North America, Europe, and Asia, enabling it to offer a comprehensive range of services including drug discovery solutions, process and pharmaceutical development services, clinical trial supplies, commercial supply of APIs, and finished dosage forms. Pyramil also offers specialized services like development and manufacturing of highly potent APIs and antibody drug conjugation. Our speaker today is Alan Davidson. Alan's the head of development at Pyramil's Grangemouth site in the UK. He has responsibility for analytical and process development teams performing method and process development, process characterization, and method validation. Alan's been working with ADCs for 7 years in both development and quality teams and has more than 24 years experience in a variety of roles spanning both large and small molecule development and quality control. Today, Alan's going to present data from a simple one factor at a time experiment, aiming to show how a small number of experiments can generate a large amount of data that is valuable to clients at key points during product development. We will be accepting questions throughout the presentation. You can send those in using the box at the lower left of your screen, and questions will be answered after Alan's talk in a brief Q&A. And this presentation will be archived on our website, contractpharma.com, for 1 year. If you're having any technical difficulties, you can type those into that question box as well, and someone on the back end here will help you out. At this point, I'll turn it over to you, Alan.
Thanks very much for the introduction and the opportunity to speak today. Welcome everyone. Good morning, good afternoon, and good evening, wherever you may be in your individual time zones. Let's get started. As Tim said in the introduction, I'm going to be talking about a one factor at a time experiment and how we can use this type of experiment to support ADC process characterization and specification setting. So just as a brief introduction and overview, I'm going to talk a little bit through Piramal Pharma Solutions in general, and specifically our bioconjugation business. I'm going to highlight how design of experiments and one factor at a time, OFAT and DOE for short, play a critical role in process development and process characterization, and highlight this using a case study where we perform a simple experiment where we're varying a T-cep to MAB ratio as the single factor, and we present some data on some typical product critical quality attributes, and really highlight how such a simple experiment in this can play an important role in supporting the definition of acceptable operating ranges and product specifications, and really just emphasize how it's possible to generate a large quantity of data from a minimal number of experiments. So for those of you that aren't familiar with Piramal Pharma Solutions and Grangemouth in particular, we're a world-leading conjugation CDMO with over 15 years experience in antibody drug conjugation and bioconjugation. Should make that clear. It's not just cytotoxic ADCs that we work with. We've capability on site to support end-to-end services from proof of concept, milligram scale conjugations, through to bulk drug substance, commercial manufacturing. We have a very highly skilled workforce across a number of key units. I'm proud to be able to say that we're recognized this year by World ADC, Hanson Wade, as the best contract manufacturing provider for ADCs. So again, just to highlight, Piramal Pharma Solutions are really one part of the wider Pyramil organization, which is a global conglomerate. We have a very diverse portfolio of sites located. It's a very east-west divide. A number of sites in the US, Canada, and Europe, and a similar number of sites in Asia. These span everything from discovery services through to formulation development, API manufacturing, antibody drug conjugates, which we'll talk more on detail, and fill finish services. And as I said in the previous slide, we really offer an end-to-end service from discovery all the way through to commercial, and where possible we try and integrate numbers of those different business units. For example, formulation can be supported in multiple sites in India for API and oral solid dose formulations, and obviously we'll talk specifically on ADCs, which are very well connected through HP API facilities in the US, conjugation here in the UK, and fill finish in the US. So really, we have a real distinct offering, particularly in the oncology arena, with we're an organisation that has a great focus here with over 70 different programs, client programs currently running in over 25 different types of cancer, and 8 of these programs are actually integrated between multiple sites in the Pyramil organisation. Obviously, these are spanning everything from colorectal, prostate, myeloma, thyroid, stomach cancers. So, and they span both clinical and commercial products. So, why Pyramil? Pyramil, we have a very customer and patient centric focus, and this is combined with a history and a track record in both development and manufacturing, supported by world-class teams across the organisation and individual business units. We're very differentiated, supporting a number of different classes of products. We're integrated across multiple sites. We have a very strong track record in quality. And really, as emphasised, we have a world-class team, over 650 scientists, and 300 plus patents with a focus on innovation across a number of sites. We'll talk more specifically about the ADC integrated offering. As I mentioned earlier, we have the capability in the US and Canada to manufacture the solid payload, which is typically used in the small molecule component in the conjugation. So high potent API services are offered in Riverview, and just outside Detroit, and we can also offer non-cytotoxic compound manufacture in Aurora in Canada. And I emphasise this because so many more of the payloads that are used in ADCs and bioconjugates in general now aren't all cytotoxic in nature. For example, use in immune stimulating antibody conjugates. So the centre of the conjugation piece really happens here in Grangemouth in the UK, and we've really been involved as a conjugation site for over 15 years now. I think we were the first contract manufacturer to actually enter this space, during which time we've manufactured well over 1,000 batches at a significant gram scale, including over 500 at GMP, and that number is constantly changing from week to week, month to month. The conjugation happens here, all the development activities, also in the formulation happen here in the UK, and we produce the bulk drug substance, which can then be onward processed at our Lexington facility in the US, either as a liquid fill or as a lyo fill formulation. So I'm going to move quite quickly from more of the sales pitch really, in terms of what Pyramil can do to the actual the nitty-gritty of the presentation and the case study. So I'm going to talk first of all a little bit about design of experiments and one factor at a time, really how those fit into development of an ADC compound. So really, before talking about OFAT, I think we really have to talk about DOE. And really, why use DOE? When do you use it? Well, it's really used both in early development, particularly during conjugation process development, and then again later to support process characterization. For those who don't know much about DOE, it's a statistical tool that allows the user to determine relationships between factors, i.e. manufacturing conditions, that affect the outputs of a process. So this is a critical tool for us when developing or characterizing a process, as it allows the assessment and or influence of multiple factors concurrently. For an example, when assessing a conjugation process, there are many different parameters that have the potential to impact on product quality, and to assess them individually, i.e. one factor at a time, is often far too time consuming. It also doesn't give you any information on interactions between factors that can significantly influence output. So DOE is used routinely at Pyramil when really identifying the early design space as to where the conjugation reaction happens. What are the key factors that influence and control that conjugation reaction? We also use it routinely in development of analytical methods, and then again we use it at later phase to support process characterization, where we're really targeting a detailed understanding of that design space in support of building towards process validation in the manufacturing environment. So what are the limitations of DOE, especially versus one factor at a time? There really aren't many, but we have seen difficulties in interpretation of some critical factors where the response, i.e. the analytical method, has an inherent variability. For example, this may be a biological potency assay, and you may be restricted within the DOE to generating a relatively low number of levels, which then may need augmentation once you've actually done your DOE study, particularly when you're assessing potential critical process parameters. So why use OFAT? Well, when it can be shown from a DOE activity that a single factor, a single process parameter plays a much larger role in controlling or affecting a specific response, i.e. a critical quality attribute of the product. So it's really to supplement data that's generated from a DOE. And as I mentioned on the previous slide, there may be a known analytical variability, in particular a biological potency assay. So the interpretation could be influenced by noise. That's effectively the center point of the DOE. And the need for analytical replication may be very high, and may be very difficult to obtain that volume of data through a DOE. It just may be very difficult to generate that data using an experiment or a type of analytical assay that generates it's not very high throughput. So really, it can also support where there's a need to understand the direct correlation. And the example we're going to talk about here is the relationship between drug to antibody ratio and potency, which is really the bulk of the key parts of the case study that we're going to talk through. So the case study focuses on the relationship between a TSEP to MAB ratio and the influence on the some critical quality attributes of a product. So for the user that aren't so familiar with antibody drug conjugation, a typical one format of ADC conjugation is a cysteine conjugation, where you target the use of interchain cysteine amino acids that hold the antibody together. And we do so by adding a reducing agent. In this case, it's TSEP. And we control the reaction by applying a known fixed charge of the TSEP to the antibody. And that allows us to control the amount of drug that can be conjugated to the antibody. So for approximately every mole equivalent of TSEP that we add to the antibody, that breaks in a single disulfide bond and creates two sulfhydryl groups. So for every bond broken, you have the potential to conjugate two drugs. So what we end up seeing is an even distribution of drug loading. So from 0, so intact antibody, 2 drugs, 4 drugs, 6 drugs, and 8 drugs. And that's what's pictorially represented on this slide. And as the slide indicates, if you target a drug-to-antibody ratio of 4, you'll probably have more of those generated in different forms than there are of the other species. So what you end up with is a very nice distribution. So we performed this experiment on 9 individual mini conjugations by varying the TSEP to MAB ratio between a ratio of 1.48 and 2.90. And the goal for us is to show how some key analytical assays vary across the TSEP to MAB ratio, focusing on correlations and distributions between some critical quality attribute assays. And we're going to highlight how this data can be used in specification setting and understanding the AORs of the acceptable operating ranges of the individual process, this specific processing parameter. And we can show how a large volume of data can be generated from one small set of experiments. So to do this, this slide highlights the number of steps that were involved in the experiment. So first thing we did was to buffer exchange the antibody into a chosen buffer. So we did this by exchanging into 20 millimolar sodium acetate 4% sucrose formulation. We then pH adjusted to increase the pH to 7.3 using a Tris EDTA buffer. That got us to an appropriate pH to perform the reduction reaction. And we performed the reduction using 1 millimolar TSEP using a controlled temperature and by varying the TSEP equivalents from 1.48 through to 2.9. So on completion of that reduction, we then performed the conjugation using BC-MMEA, so monomethyl auristatin with a maleimide linker at 7 equivalents for 45 minutes. And we then quenched the reaction using 30 millimolar N-acetyl cysteine, again for 45 minutes. Post-conjugation and quenched, we then desalted back into 20 millimolar acetate buffer. And the purpose of that desalt is really to remove the excess drug and quench reagents. So we then moved on to the analysis. We generated our 9 conjugates, and then we focused on analysing some of the critical quality attributes of the product. So first assay we did was a hydrophobic interaction chromatography assay. And what we're looking at here is how does the drug-to-antibody ratio vary with the TSEP to MAB ratio. And as you can see here, there's a nice pictorial representation, a nice chromatogram of our DAR4 showing 0 drugs, 2 drugs, 4 drugs, 6 drugs, and 8 drugs. And we can integrate those, and that gives us an average DAR by assessing the contribution from each of those individual peaks. To do this, we just used some platform conditions, tosyl-butyl NPR column, standard ammonium sulfate phosphate buffers, buffer B with IPA to help elute some of the higher drug load species, using a standard gradient that we would typically apply for when developing a method for a new product. So how does the DAR vary? Now you can see from this graph alone that there's a noticeable correlation between the DAR and the TSEP equivalents, increasing proportionally with the increase in TSEP charge. All of the data here met expectations, with the exception of sample 2. The astute among you will notice that there's only 8 results listed here. Now, we had a problem with sample 2, which was the TSEP to MAB ratio of 1.66, and we've excluded that from all the following data, and we'll show you why on the following slide. So, as you see here, this chromatogram looks significantly different from the one in the previous slide. Now, this was obviously an experimental error. Almost certainly there was insufficient drug added at the conjugation stage, which meant that we didn't occupy all of those self-hydryl groups. So we've ended up with not a nice even distribution, 0, 2, 4, 6, and 8. We've ended up with some odd numbers here, some 1 and 3. And this sample gave us an average drug-to-antibody ratio of only 1.85. And this is consistent with the data from the potency assay that we'll present later, and also non-reduced CE-SDS. This is just an example of something that can go wrong, particularly when you're performing mini conjugations with relatively small amounts of material. So what does the correlation between the TSEP-MAB ratio and the drug-to-antibody ratio look like? Well, here's the figure that actually displays this. So as you can see here, there's an excellent linear correlation between the two, with an R-squared value of greater than 0.99, which shows how good a correlation it actually was. Now, using the equation of the straight line here, we can typically expect that targeting a DAR of 4 that with an early phase specification, we would typically expect a specification of 4 plus or minus 0.5, so a specification of 3.5 to 4.5. And by rearranging the equation of the straight line, we can determine exactly what those specifications equate to. So you can see here in the table on the lower left side that a specification of 3.5 equates to a TSEP to MAB equivalent of 1.9, and at the upper specification, 2.51. So this slide really presents the same data, but what we've done here is present the specification limits. Now, effectively, these specification limits are equivalent to the acceptable operating range of this process parameter, because you can't operate outside of these. So the figure on the left shows graphically the lower specification limit of 3.5 and the upper of 4.5. And the graph on the right displays the normal operating range, which is typically approximated based on the typical tolerances of balances, etc., that are used to control those charge additions of input antibody and TSEP. And you can see here that the normal operating range is very narrow. It typically can be controlled very, very well. Now, when you drill into the data in a little bit more detail, and you start to plot how all the individual species, so your DAR 0, 2, 4, 6, and 8 vary across the range of TSEP to MAB ratios, you'll see here that there's some very noticeable patterns. For example, you can see that the DAR 2 species in orange and the DAR 6 species in the gold color, these all follow very, very good linear correlations, with all the other species following good second-order polynomial fits. And when we look at this data and focusing on the two lines that have been added to this represent the upper and lower specification limits, you can really start to see how within a specification your product can look very different from the lower end of the specification to the upper end. We highlight this here. the distribution is highly variable within these two limits, specifically for DAR 2, 6, and 8 species. And all of these species can be individually modelled using the line formula for the linear fits or the second order polynomials. And by doing so, it's possible for us to actually numerically determine what that change from the lower specification limit to the upper specification limits here are. And you can see here in the table that for DAR 2 species, there's nearly a 15% change between the lower spec limit and the upper spec limit, with quite significant changes for DAR 6 and DAR 8 as well. Now this is important because obviously as you develop more knowledge of the product, you may be thinking more about your product and your specification, and starting to ask the questions, how do you control these, and should you control them? Which is exactly what we're talking about on this slide. So, do you control the distribution? Unconjugated antibody, the percentage of DAR 0s, it's always of interest to regulators, and there's more being done to understand the influence that the unconjugated antibody can actually play, whether that's is it competing against your conjugated drug for uptake in the cell, or does it have any other influence on the quality of your product? But there's typically a focus on controlling your level of unconjugated antibody. So, asking the question here, do you assign limits for DAR 0? Noted here that a 5% maximum limit would reduce the AOR at the lower end. As you can see here, highlighted that even at 1.9 T-septimab equivalents, you're at a percentage of 6.1. So to control, for example, hypothetically, to control the DAR 0 at a maximum of 5%, you would have to amend the AOR at the lower end of the specification. Ask the question here, do you assign limits to DAR 2, 6, and 8? Does all show significant change? Now, early phase, you're unlikely to do that, as you still don't have much product knowledge, but we'll come back and discuss this a little bit more once we've actually looked at the non-reducing CE data and the potency by cell killing. Before doing so, I'm just going to touch on one of the other key critical quality attributes that you're always interested in when performing conjugation or anything involving ADCs. You're always concerned about, is your product, does it have the propensity to aggregate? And in this case, we did this assessment using some platform conditions. You'll see the figure on the right here is a representation of the sample with a drug to antibody ratio of 4, and you can see on the figure high molecular weight and the low molecular weight species, with the low molecular weight being visible as unresolved fragment on the tail of the main peak, and as a separate smaller species. So here's some platform conditions here to make this assessment, and the data is presented on the following slide. Now here, we didn't really see much of a correlation between the T-septimab ratio and any level of specifically for aggregation. Obviously, adding relatively large amounts of hydrophobic drug to an antibody can cause aggregation. In this case, we didn't really see anything. We only saw a change of around 0.5%, and there's no real visible trend between the T-septimab ratio sample at 1.48 versus the one at 2.9. So we're not going to discuss size exclusion and levels of aggregation in any more details. There's much more interesting data to be sought. So one of the other assays that really gives us some interesting data here, and it gives us an understanding and supports the data from the HIC that we've just presented, is non-reducing capillary electrophoresis, SDS. And for those of you that don't know so much about this assay, the way this assay works is to bind sodium dodecyl sulfate to the antibody to give it a uniform charge, and then to alkylate the sample to prevent disulfide bond interactions. The samples are then separated on a capillary column, and they're separated on the basis of the molecular weight. And the changes in molecule reflect the level of reduction that's been performed, i.e. the number of TSEP equivalents that we've been discussing, and equate to a measure of that part of the manufacturing process. Now we do this using a SciEx P800 instrument, and again we used some platform conditions here, so we didn't do any development work prior to performing this experiment. And you'll see on this slide, this is an electropherograph or 2 electropherograms overlaid. The blue trace represents the lowest TSEP mAb sample with a DAR of 2.8, and the black trace represents the conjugate with a average DAR of 5.1. Now you can see here with the increase in TSEP equivalents that the smaller species, your light chain, your heavy chain, and your heavy light species are all increasing. The light chain and the heavy chain increasing significantly from lowest drug to antibody ratio to highest, and those correspond also with a drop in significant drops in the heavy light and intact antibody species, and you can see those in more detail on this slide, where we present the distribution of all the species in the same ways we did to the hydrophobic interaction data on the earlier slides. So again, you can see that a number of the species follow linear correlations, and the remaining species all follow very good second-order polynomial fits. Specifically here, the light chain, the heavy chain, and the heavy light all have very good linear correlations, and you can actually see that the percentage of heavy light is dropping proportionally to the increase in the percentage of heavy chain. So you can basically assume here that heavy light is been broken to form more heavy chain and more light chain. And again, similar to the HIC data, this slide really presents the lower specification limit, or the lower AOR value, 1.9, and the upper AOR value of 2.51, and it shows just how variable the profile and the distribution of species are within your specification range. And again, we've done the same experiment here, where we've taken the individual line formula and we've calculated what the percentage change is across the AOR. And as you see here, there's a significant change in the heavy chain species and the heavy light, with 9% heavy chain change and a drop in heavy light of over 11%. And again, this graph here is a reflection of what the NOR looks like. So the same NOR displayed earlier shows that even within the NOR, you can see that there is a significant change, particularly for those two species, heavy chain and heavy light, as highlighted there. So light chain, heavy chains show minimal variation. They're fairly well controlled across the NOR, and relatively so across the specification range. So what sort of specification would you apply at an early phase here? You're likely to use some appropriate wording such as conforms to reference, but the key thing is, conforms to reference has to have a definition. It has to mean something. So you need to define that. And how do you define that? This is where you have to be really careful, because obviously you've highlighted here that there's a significant change across your acceptable operating range, and there's a relatively sizable change just within your NOR. So you must be really careful in terms of assigning these as a distribution of species. I would definitely suggest not doing that at an early phase. Maybe something that you start to consider by the time you get closer to later phase, but all of this should be driven by understanding of the process and a history and a data sets associated with batch manufacturing, and you must also consider the analytical variability. So what is your expected analytical variability through intermediate precision? So changing your operator, changing your capillary, changing your instrument, changing days. All of these factors need to be understood before you consider applying some distribution criteria. So you may choose to have a more woolly definition of conforms to reference, but it still has to be meaningful, and it should be clearly explained. This really takes us into one of the key things that drug to antibody ratio drives is the potency of the molecule. So you can expect to see that as you increase the drug load, the average DAR, you should expect to see an increase in potency. And how do we do that? We measure those by an in vitro cell-based assay. And you can see here the graphic on the upper right-hand side. It's really a representation of how an ADC works. So the antibody binds to the antigen that's ideally overexpressed on the surface of the tumour cell. It's then absorbed, internalized into the cell. And in the case of the IgG1 BCMMEA conjugate that we've manufactured here, it relies on a protease within the cell to act on cathepsin, to act on the linker and release the drug, which generates the cell-killing effect. And we'll talk about a little bit more on the following slides. The format of the assay we've used here is we use a frozen cell format, where in this case we've taken a ready banked working cell bank, and we've seeded cells at approximately 8,500 cells per well. We've then established a working range, a dilution scheme, where we expect to see the potency effect. We allow the cells to recover overnight at 37 degrees before applying the drug, the dose dilutions, and incubating for approximately 6 to 8 hours. And then we read the metabolic response using luminescence, using cell titer glow reagents. And we did this by only performing 2 experiments. So the first experiment we did was to take the average, the center point of the study, the DAR4 sample, and also we included the lowest potency sample and the highest potency sample in this experiment, and we performed an extended 24-point dilution series. And what that did was it helped us identify the point at which the product had its linear portion. So where we could see the potency effect, we could see cell killing on the lower asymptote and no cell killing on the upper asymptote. And once we had that data, we then performed a second experiment using a reduced 8-point dilution series. And I should highlight here that we typically use an 11-point dilution series using a well-characterized reference standard. But in this case, we used the center point, the DAR4 sample, to measure all the other potencies relative to this sample. And here you see the initial 24-point experiment that was used. This is just a partial representation. It's not the full curve, but you can see on the left the upper asymptote and on the right the lower asymptote. with the linear portion of the potency, the sigmoidal curve in the centre. And this allowed us to assign our starting point for the main experiment. And here you see a very, very busy, these are the observations that you could see here. So you can see on the upper right-hand side, the DAR 2.8, average DAR sample in the green, and in the orange, the highest DAR sample at 5.1. And in the middle, somewhere along there, the DAR 4.0. There's two things to really observe here, is that with the increase in potency, you see a shift from right to left, which is expected, and this is how you really measure your relative potency. But one key other observation is, as you can see on the right-hand side, that on the lower asymptote, there's quite a big difference between the highest drug load sample and the lowest, and this is also a representation of the percentage kill. And what you're seeing here is that for the drug to be effective, the drug-to-antibody ratio really has to meet a certain threshold with more free antibody and a higher percentage of DAR2 than the DAR 2.1, which is much less effective in delivering the minimum threshold required to actually achieve the cytotoxicity. And the higher luminescence at the lower asymptote is really indicating that the fewer of the cell population have received that threshold dose to actually perform, that actually influence the cytotoxicity. Now this is obviously a graphical representation. Now we can determine the relative potencies of all of these using parallel line analysis, using statistics. And what we've done here is display the HIC data, so the average DAR data, against the corresponding relative potency data. You see there the DAR4 sample has been assigned 100% relative potency, and all of the other species measured, all the other conjugates measured from this. Typically, this type of potency has a geometric COV of less than 10%, with less than 3% bias over a concentration range of 50 to 200%. And the potency values between approximately DAR of 3.8 and 4.3 are within the natural variability of the assay, which would typically deliver a potency of between 80 and 125%. But you also see that there's potential to increase the potency twofold by a corresponding twofold increase in drug load. So the highest DAR sample here at 2.9 is at 200%. And those can be represented again by a correlation plot. So if you plot the TSEP to MAB ratio against the relative potency, you see some excellent linear correlations. And again on the left, these are representations of the acceptable operating ranges or the specifications of equivalent to a specification of 3.5 to 4.5, and on the right hand side the normal operating range of the assay. But even within the normal operating range, you're really looking at approximately a 10% change in relative potency over this range. And again, what we've done here is we've actually plotted the drug-to-antibody ratio against the relative potency, and the correlation has actually improved. It's actually improved on the TSEP to MAB ratio versus relative potency. And again, by rearranging the equation of the straight line, the lower specification, or AOR, and the upper spec, or other end of the AOR, really equate to relative potencies of roughly between 67 and 146%. Now, it's typical that an early phase specification for this type of molecule would typically have a specification of maybe between 50 and 150% relative potency. But I think it's clear here that for this conjugate that we've made, that it's unlikely that this specification could be supported. As you can see that the upper specification range is already at approximately 146%. And obviously, before considering setting that as a specification, you need to fully understand the performance of the cell-based assay, particularly the precision, the accuracy, linearity and bias, etc. So really, we've highlighted here on the previous sets of data, we've looked at drug-to-antibody ratio, we've looked at the distribution by non-reducing CE, we've looked at potency. So as we've said earlier, a DAR of 4 plus or minus 0.5 would be very typical for an early phase process. You would typically also expect to report the level of unconjugated antibody. I would certainly advise not setting a specification at early phase, but to report the result and start to build a body of data. Non-reducing CE, you'd have a specification of conforms to reference standard, but you really, as I said earlier, you need to make a clear definition of what that means, and whether that's a rank order of species with some sort of reflection that within the specification range you may see a change in distribution of area percents, particularly for the heavy chain and heavy light species. I'd certainly recommend that, but it may be very wide at this stage. And absolutely as a minimum, you should be trained in all of this data and starting to build a body of data to help you support specification setting at later phase. Now the potency, subject to the additional analytical data assessing the performance of the assay, 50 to 200%, it's very wide, but it may be appropriate at this early level. And obviously the performance of the assay may allow you to tighten that. Alternatively, if you have to control that potency more, this would correspondingly impact on the DAR specification and the AOR that you're setting for the product. Now, what do you consider for late phase? You'd really expect that the DAR should be able to be controlled much tighter than that highlighted for early phase. The control is typically very good for this type of conjugation, and the expectation would be there that with additional batch data, process capability knowledge, you would be able to tighten that. But in this case, there is no substitute for data. It's all about batch data and batch history and understanding of what the process capability actually is. Now, percentage unconjugated antibody, if this is something the regulatory agencies are pushing for, may be asked to set upper and lower limits. And again, that should only be done once you have a better understanding of the manufacturing process, process capability, and batch data. Again, no substitutes. The non-reducing CE-SDS conforms to reference standard is still an appropriate specification, but that definition may have to become much clearer, and there may be a push both for the HIC assay as well as the non-reducing CE to set some clear expectations on what the distribution and the ranges for these individual species are. And again, that's got to be based on process capability and analytical method understanding and variability. Now, potency, again, subject to that additional data which can be generated during qualification or validation. The method capability understanding is absolutely critical, and that will define the degree of replication that the assay format needs to form. And these are key statistical measures that you need to assess during early phase qualification and late stage validation, where you need to look at the methods, capability, the CPM, and also the probability of an out of specification event. And the best way of controlling, improving method capability and reducing the likelihood of an out of specification event is to increase the replication. Now, if you increase the replication, i.e. the number of events you have to perform to generate a reportable result, that has other implications on throughput. And then you need to consider how you're going to apply that when you're performing process characterization. So again, considerations for late phase. How do you define the AORs? The data from a study such as this could support the AOR, not driven by the chosen specifications, specifically for the T-cep to MAB ratio and any other assay data that's presented. Now the AOR will tighten depending on a requirement to minimize the amount of unconjugated antibody present or any other species present, and that has to be factored into how you apply restrictions around control of species so that you can help support AOR definition. I'm going to summarize the experiments that have been performed now. So we performed one scouting experiment prior to generating the 9 mini-conjugations, and that was supported within the PD team, the process development team here, just by looking at the DAR relationship using hydrophobic interaction chromatography and size exclusion just to understand aggregation. That experiment allowed us to perform the 9 conjugates that we've been discussing in this case study, and then we added analytical methods to that. We added non-reducing CE and also the potency. And to make it clear, we did no optimization for any of these. These are just off-the-shelf platform methods that were utilized. The T-cep to MAB ratio was supported by an in vitro cytotoxicity assay. And again, as I've said earlier, using a banked frozen receptor line, receptor cell line. And we only performed 2 experiments here. And we were able to do that just based on our approach. We've got a very strong platform approach to developing cell-based assays. We've got a very good understanding of standard incubation, cell numbers, incubation times that are generally specific to different drug species as well. And really just want to emphasize just how little effort was required to generate all of this data. And I've not even presented all of the different correlations between some of the individual assays, but it highlights that a huge amount of data can be generated just within 3 assays, supporting 9 individual mini-conjugations. So by varying this single one factor, we're able to develop a huge amount of understanding of correlations between changes in species for critical quality attributes. We've got a really early stage understanding of how the potency changes across changing that factor, and this really helps use very early in product development and gives you valuable insights into how you may want to control a process. And we're starting to conclude the slides here, and just really want to say thanks to the process development and analytical development team, to Owen and Triana, who performed all the mini conjugations and some of the analysis, to Zav, who helped Owen and Triana design the experiments, and to the analytical development team, Graham, who performed the CE, Eric and Lauren, who performed the potency experiments, and Robert, who's the bioassay lead, and really has helped establish this format and reviewed some of the slides specifically around the potency testing. So with thanks, I really like to say thank you for listening in, everybody, and I guess that then opens the floor to questions.
Yeah, great. Thank you so much, Alan. A really informative and detailed presentation there. Like Alan said, we do have time for some questions, so we'll open the floor up. If you do have a question of Alan, now is the time to type it into that box there at the lower left of your screen. So let's get through a handful of these here. First question. In the experiment, why have you targeted a drug-to-antibody ratio of 4? Why not target a higher DAR, and what might be the implications in doing so?
Yeah, no, it's a very valid question. Obviously, I think it's industry is very well accepted that with this type of drug linker, VC-MMAE, that the optimum DAR is a DAR of 4. That's been shown, within the clinic, and it's a case for commercial products, in case of more than one commercial product that use this payload and with a target DAR of 4. In terms of why not a higher DAR, the implications really are more. You can see here the change in potency in vitro increases, but that may not be the case in vivo. It's very well understood that with a significant increase in higher DAR species, they become very hydrophobic, and within the body, some of those actually have a much shorter half-life than your lower DAR species, and can actually be cleared much faster by doing so. In some cases, your higher drug to antibody samples also are more prone to aggregation, and therefore less stable. And obviously, aggregation is something that has to be controlled very carefully for parenteral compounds such as this, where you want to minimise any immune response. But yeah, no, it's a very valid question.
Okay, great, thanks. Let's see here. Can you be more specific about when during development you would perform an experiment such as this?
Yeah. I think there's two opportunities to perform this experiment, and you may do it in two different formats. I think the case study we've highlighted here could be one that could be used during early development. During conjugation development, which is one of probably the early activities when developing a conjugation purification process, is that you could apply this experiment to really start to give a much more detailed understanding of how some of the key or critical quality attributes of the product are affected by an important factor such as your T-cell to MAB ratio. And then the second maybe iteration of this experiment would be to support process characterization, perhaps to augment data that's been generated during the DOE, and really help you understand in much more detail the impact of that one factor, but to also generate a lot of data that supports your specification setting and your AOR determination. So I would really could see this experiment be performed in two different, one in a very early phase format and one in a much more detailed late phase when again the process is more developed, it's well understood, and the same can be said for the individual analytical methods.
Okay, let's see, moving on here. In the potency experiment, what would be the impact of the presence of free drug on the reportable result? And have you tested these samples for free drug, and what species would you typically test for?
Okay, right. Well, I can say we've not tested these samples for free drug. That was one experiment we would like to have performed, but we minimal amounts of material, and we didn't have the opportunity to spend some time to actually develop that method, which would require a little bit more development in this case. But what we did do to mitigate this, I should really come back to the first question actually, is that the impact could be quite significant on the potency experiment, as the free payload is highly toxic, and it would do so in a non-specific way. So any presence of any free drug species here would cause a noticeable shift in potency. So to that end, what we actually did was we did a second desalt before performing the potency experiments. So in one of the early slides, you saw that the final stage post-conjugation and quench was at a desalt. So we repeated that to be confident that we were removing any excess payloads. My notes. Yeah, the other part of that question was, what are the species? So yeah, we would you've really got two main species to test for from this experiment. You would have the free payload or the free drug linker, so your VC-MMEA, but you would also quench for the test for the quenched form of that as well. It would typically be the quenched form you would expect to see, but you should also be looking out for the actual drug linker itself, and it's certainly worth also including the actual drug without any linker, as it is significantly more potent than the drug linker or the quenched form. But yeah, no, all three should be tested for.
All right, thank you. Let's see here. You present a couple of slides on aggregation by SEC HPLC, but don't discuss further as there was no correlation. Would you typically expect to see a correlation between DAR and aggregation?
You may do, yeah, you may. I think I alluded to this towards the end of the talk as well. Yeah, obviously you are actually conjugating a relatively very hydrophobic molecule to an antibody. So obviously the more payload you add to the antibody, often the increase, there's more of an increase in propensity for aggregation, aggregate formation, and that's even more true in other types of drug, particularly some of the methansines or pyrrolobenzodiazepines. There's often the need in some of those processes for a chromatography step between conjugation and purification to actually remove some aggregate species, and yeah, you may see a direct correlation between the two factors, but in the case of the experiment we've highlighted here, there really wasn't any, which I think if this was a real product, would be quite a nice place to be.
Okay, great. Thanks, Alan. Let's see here. We are running over time, so we're going to take one more question before wrapping things up. And it asks, in the non-reduced CE-SDS data, would you not expect to be able to get better resolution between some of the higher molecular weight species?
Yeah, no, definitely. I think this is a reflection of the fact that we didn't perform any development or any trials prior to running these experiments, and most likely what we've seen here is we've overloaded on the capillary. There's more protein going onto the capillary than we would typically expect to see. So we'd probably in this case look to modify, if we're developing this as an assay, we'd look to modify either one or both of the injection time or the injection voltage to achieve a better load and correspondingly better resolution. But yeah, no, it's a valid question.
All right, terrific. All right, well, that's going to go ahead and conclude today's presentation. If we didn't get to your question, Alan will respond to you directly. He has your details, and we'll follow up. I'd like to thank you again, Alan, for a terrific presentation, and thanks again to today's sponsor, Piramal Pharma Solutions, and thank you, everyone in the audience, for your attendance and participation. As I mentioned earlier, this session will be archived for 1 year on our website, contractpharma.com. Thanks very much for joining us, and have a great rest of your day.